There we go. I want to welcome all of you to the Centers of Excellence, Iron and Heme Disorders at the University of Utah virtual seminar series. This is our second, maybe almost third year because of COVID of doing these things, not in person. And I'm super grateful that we at least have this to be able to see each other and share our science and brainstorm with each other about great ideas of science and learn from people who are really pushing the forefront. And today we have Amit Reddi, who's going to be speaking to us. Before I introduce him, I want to just share my screen with you and remind you of some funding opportunities that are available. So, all right. There we go. This is our website, which is the CIHD.cors.utah.edu website for the Center for Iron and Heme Disorders. And if you go to this website, you can click on this funding opportunity and click grants here. We here at the University of Utah have a pilot and feasibility program opportunity, and I highly recommend you watch this site for chances to compete for funding opportunities here at Utah. Similarly, if you click on this link, you will find funding opportunities that's for all the different centers in non-malignant hematology. And what they have are two types of funding opportunities. They're called type A grants and type B grants. The type A grants are essentially banking core credits at a core that you might utilize. And so that's not just at Utah, but it's at any core. But at Utah, you could use the iron and heme core, you could be using the metabolomics core, and these are just a one-page essentially application to try and bank some funds up to $12,000 at a core that you might use that is sponsored by these centers of excellence. The type B grants right down here have up to a $75,000 award, and we have a call that's due. It's actually due next week, but they're not a super long application either. And we're really looking for, innovative pilot research in non-malignant hematology. And these types of funds really work and are a good amount of money to be able to generate data for applying for extramural funding. So if you're in that arena, I highly recommend you check out these websites, and there's all the instructions here. And if you go down to the bottom here, there's a little place where you deposit your grant application, and you can actually look below to see some of the pilot and feasibility grants that have been supported by the Centers of Excellence in non-malignant hematology. So I'm going to stop sharing there, and then I'm going to just quickly introduce Amit. Amit comes to us from Georgia Tech University. Is it called Georgia Tech University? I don't know.
Georgia Institute of Technology.
There's not a university on there. That's right. Amit got his bachelor's degree from Carleton College, which I keep being stunned about the number of people I know who went to Carleton. When I read that, I'm like, wow, okay, there's a lot here at Utah. Mm-hmm. Okay, and then he went on to get his Ph.D. at Columbia University in the laboratory of Brian Gibney. He did a postdoctoral fellowship in the laboratory of Valerie Culata. So many of us who've been in the iron and metals field for many, many years and using yeast as a model system have been acquainted with Val's research, and Amit was a very prominent part of that research. He then moved on to a faculty position at Georgia Tech, where he is now an associate professor in the School of Chemistry and Biochemistry. And today he's going to talk to us, and the title of his talk is Illuminating Heme Trafficking and Signaling in Health and Disease. I'm going to turn the time over to Amit, but want to recommend to you that everybody silence themselves while Amit speaks, and then I can invite you in to ask questions. Amit has agreed to answer questions throughout the talk. So if you have them, you can just populate them into the chat so I can see them, and then I can invite you at appropriate pauses if you would. And so if you will, all please silence yourselves except for Amit. That would be great, and we'll turn the time over to him. Thank you so much, Amit, for coming to speak with us today.
Yeah, thank you, Diane. It's absolutely great to be here, even virtually. And I really want to thank Diane for the invitation, and it's really nice that we're able to come together and just talk science, whether there's a pandemic or not. I really appreciate that. So for those that don't know me, my laboratory is interested in the cell biology and biochemistry of metals and metalloproteins. And so we're really interested in fundamental questions related to how cells manage metal ions, and in particular iron and heme iron. But we're also interested in the impact of metals and metalloproteins on redox homeostasis. But for today's talk, I'm going to focus it on our work related to heme and heme cell biology. I'm going to, okay. So I want to start by just going over the types of questions our lab is interested in. Hold on, I'm just going to pick out a laser pointer here. Okay. So most cells on the face of the planet, they require heme. They're either able to make it or bring it in or do a combination of both. And the questions we're interested in are related to how cells traffic and manage heme. So in a eukaryote, heme is made in the mitochondria, and it's got to be transported and trafficked out of the mitochondria to resident hemoproteins in virtually every subcellular compartment. And it's not clear how heme is mobilized to all of these different resident hemoproteins. When you think about the trafficking challenge, heme is a hydrophobic molecule. It's redox active. It's going to stick to things, and it's going to potentially catalyze deleterious redox reactions. And so you might think, without even knowing anything about the system, that there may be dedicated mechanisms to help safely shepherd heme from point A to point B. And so a fundamental question we're interested in are, what are the factors responsible for mobilizing and trafficking heme? Another related question is how dynamic is this mobilization? Over what timescales does it occur? What initiates its mobilization? What gates the ability of heme to get from point A to point B? And another question we're interested in is this question of heme signaling. How are heme signals initiated, transmitted, and integrated into physiology? It's becoming increasingly clear over the last decade or more that there are proteins in cells that can be allosterically regulated by heme to control their function. And so we're interested in understanding how heme signaling works. Where are the signaling pools of heme coming from to control these downstream processes? And how, again, how do you safely mobilize a heme signal without negatively impacting metabolism? One really useful conceptual paradigm that helps us think about these problems is envisioning all of the heme in a cell as some combination between exchange inert and exchange labile heme. So exchange inert heme is a pool of heme that you might consider to be associated with high affinity heme enzymes, where that heme may be bound and buried in some enzyme active site that's not competent for exchange with other factors. And so we might, but for the purposes of this discussion, we're more interested in what I call this exchange labile pool that may be more relevant for trafficking and signaling. And this might be heme that's bound to some factor that for which there's very little kinetic barrier towards exchange to other factors. And it's this pool that I'd argue may be relevant for trafficking and signaling, and is the pool that we're most interested in deciphering its nature, its speciation, and what controls it. So when we started, our lab decided to focus on problems related to heme trafficking and signaling by first developing reagents that would help us sense and image this labile pool of heme. And so what we did was we designed a genetically encoded sensor that could image this labile heme pool. The way it works is that there's a heme binding moiety fused to GFP. Heme binding to the heme binding moiety quenches GFP fluorescence through an energy transfer mechanism. And there's a second fluorophore whose fluorescence is insensitive to the presence of heme. And we're ratioing the change in GFP fluorescence to this red fluorescent molecule that is insensitive to the presence of heme. And so you can make this sensor, you can purify it, it behaves as designed, you can titrate up heme, and you can see a robust quenching of GFP fluorescence with a relatively invariant signal change from the red fluorescent protein MK. We have, so we've made a library of these sensors. You can imagine that many different cell types might have different heme quotas and dependencies on heme. So if you're a red blood cell, you know, when you're mature, you have very high levels of heme. So that might require you to have a lower affinity heme sensor in order to image high levels of heme, and other cell types may have very limiting amounts of heme, where you might need a higher affinity sensor to image very low levels of labile heme. So we've created a library of about 50 or so variants that we now have the ability to basically plug into any cell type that we care about and image heme by using this fluorescence, this heme sensitive fluorescence response in a variety of different cell types. You know, all of the questions that I outlined in the previous slide are going to be relevant to many different types of cell types. So we're interested in microbial pathogens like mycobacteria, tuberculosis, and Candida albicans. They have very different heme requirements than eukaryote, than for example, various human cell lines. And so the point here is you have to have the right tool to image the right amount of heme in the right compartment in the right context. And so I think we've achieved that with the library of sensors we've developed. So this is just to give you a snapshot of what we are imaging when we look at heme in different cell types. So this is an example of yeast cells where we can get a characteristic ratio of our heme sensor and wild type cells. And if we heme deplete using a heme synthetic inhibitor like succinylacetone, or if we use a mutant that can't make heme, we get characteristically different fluorescence ratio. So we're really just monitoring changes in fluorescence ratio to infer, to make inferences about what's happening to this exchange labile heme pool. Importantly, we can also target the sensors to different organelles in order to get subcellular resolution on local heme pools and specific subcellular compartments. And I'll bring that point up in just a minute. So, you know, after developing this library of sensors and having you know, this expansive toolkit that we can now use in different contexts, we wanted to set about identifying and addressing the questions I brought up earlier. For example, what are the factors that regulate steady state heme distribution? You know, what are factors that regulate heme dynamics? And another question that I'll address here is what are factors that are responsible for distributing heme as it's being synthesized in real time? And so I'll quickly go through the approaches we've taken and the lessons learned from understanding what governs steady state heme distribution and I guess non-steady state heme distribution. And then I'll transition over to our more latest work, deciphering heme signaling and factors that are targets in heme signaling. So in the first 2 points, a lot of this work is already published, but I'll just sort of give you a broad overview of it so you get a sense of what we're doing and how we're doing it and some of the interesting conclusions from that work. So one of the first things that we did was, you know, although we work in many model cell lines and organisms, I'd say the workhorse organism for us is Baker's yeast. I think that many of you in the audience can appreciate that that it's a simple microbe that we can culture very easily. Two things that really stand out about it are that we have the entire knockout collection in our freezer, so it's really amenable to high throughput screens. But another really important point about Baker's yeast is it's culturable in the absence of heme. And so that's really important because that gives us a zero heme background with which to really understand heme metabolism in a way that we're not confounded by contributions from other processes. So, for example, in mammalian cells, you can't culture those cells in the complete absence of heme, otherwise they're dead. So yeast gives us a little bit more flexibility. So one of the things that we wanted to do is just screen the knockout collection and just ask the question, what regulates heme availability as read out by our sensors? We already know a lot about heme synthesis and degradation. So we know that heme is made from the condensation of glycine and succinyl-CoA in 8 enzymatic steps, ultimately to produce heme, where iron is inserted into an organic porphyrin macrocycle. And so we should, if we screen the yeast knockout collection for factors that impact heme availability, we know we're going to get factors that influence heme synthesis, and those are all already annotated. But the interesting thing that we want to pull out of the screen are what are the factors that govern heme availability, and those may be less obvious from typical gene annotations. So we can screen the knockout collection. The collection has the yeast genome has about 6,000 genes. There's about 5,000 deletion mutants that are in the haploid knockout collection. And we were able to stably transform about 4,500 of those deletion mutants with the heme sensor. And so we can pull out a number of mutants that have higher or lower heme. And this is just to give you a reference point. Shown on the left here is are all of the mutants that we screened. The y-axis is the z-score. So right in the middle, wild type is about an average strain compared to all the others, and it's got a characteristic fluorescence ratio and labile heme amount. On the extreme right, at very low z-scores, negative z-scores, is a Hem1 knockout. So that's our control for a cell that's got no heme and therefore very little or no labile heme. And so what we're really interested in are what are these mutants that have Hem1 knockout-like levels of heme availability, which tells us that there's a gross defect in the ability of these cells to make heme bioavailable. And that's the first little snippet I'll tell you about today. So as expected, our screen pulled out all of the stuff that you would expect to control heme synthesis, like pyruvate transporters, iron transporters, anything that impacts heme synthesis that we already know about came out of our screen. So that's a really good validation for the approach. But what I'll tell you is that about 80% of all of the genes of the deletion mutants we identified all impact Golgi to vacuolar protein sorting and trafficking. Many of these genes impact the formation of endosomes, and so this gave us a clue as to maybe heme is being trafficked in a way that where it's trapped in these vesicles. And they're being mobilized from these vesicles and ultimately from the vacuole. And when you disrupt any of these processes, if you disrupt vacuolar function, or if you disrupt any of these protein sorting pathways, you completely abolish the presence of labile heme in the cytosol. And so that's our working model. And so this raises some interesting questions as to can we probe vacuolar heme or heme in these endocytic compartments? And so that's what our lab has been working on for the last year and a half or so. We've been working on sensors that can withstand the relatively acidic and hostile environment of these acidic compartments in order to see if we can image heme in these compartments and see if we can test our hypothesis that that ablation or defects in these protein sorting pathways results in a buildup of heme in these endocytic vesicles and the vacuole. So that's a hypothesis we're testing right now. Another question we're interested in is, how does heme, get into these in these circulatory vesicles in the first place? How does heme get into the ER and the trans-Golgi network? And that's a point I'll address in a couple of slides in this talk.
Hey, Amit,
Yep.
I'm going to interrupt you. Will you go back one slide?
Yes.
Why?
Yeah.
Is heme in the cytosol a bad thing? Or why are you putting it in a vesicular compartment?
Yeah, it's a good question. It's an excellent question. So at the start of all this, my sense is that well, I'll tell you our first initial experiments. Our first initial experiments told us that most of the labile heme was in the cytosol. So if you probe labile heme across multiple compartments, we saw most of it was in the cytosol. And so that initially led to the thinking that, maybe the cytosol is best adapted to hold on to this, what we're referring to as this bioavailable heme pool. And so we figured, okay, there's gonna be lots of factors in the cytosol that bind and buffer heme. So after doing the screen, this sort of ran contrary to that initial thinking from that early work, is that it seems that heme is being packaged in these vesicles in a way that in a way that sequesters it, and then and the implication there is that maybe this heme has to be sequestered in a way that away from general cytosolic components in order to mitigate toxicity and that sort of thing. So really, Diane, I think the answer to your question is I think this has to happen because you want to limit heme in the cytosol to levels that maybe exceed some threshold value, maybe that exceed whatever the buffering capacity is in the cytosol. I think what we have to do is look at some of these mutants and ask the question, are any of the growth defects associated with these mutants really because of heme toxicity or not? That's something that we don't yet know. But I think part of this model is possibly that this has to happen because these vesicles provide a heme detoxification mechanism. I think that this is consistent with some of the recent work that's come out in the literature by collaborator and colleague Iqbal Hamza. Has found that through label-free imaging approaches that there are these heme foci that you can image using transient absorption spectroscopy, and that very well could be heme concentrated in these types of vesicles. But I think a lot more has to be done to understand the physiological context and the why question. Why does this have to be the case? But I think that's something we have to figure out.
One more question.
Yeah. Hi, I'm Jason Shapiro. Related question. Not sure if it's if you're going to get to it later on. And so when I think about vacuoles and endosomes and lysosomes, and also the trans-Golgi, they're kind of two different biological processes that I think of, at least in mammalian cells. One is the recycling component. And so we have methods to recycle ferritin and other heme through the lysosome, and it's dependent on the autophagy mechanisms. Do you see the same thing with heme? Do the HEGs and other autophagy molecules are required for the lysosomal and vacuolar heme levels? And then for the trans-Golgi, do you think it's involved in like loading heme into folding glycoproteins and things like that?
Yeah, it's a great question. To address the first part, the answer is yes. So genetically, and everything that impacts a lot of the genes that impact any of these protein sorting pathways, vacuolar acidification, autophagy as well, it results in this low cytosolic labile heme phenotype that I'm seeing. So I think all of these pathways are connected. And so the question now is, is really is one of, can we do the epistasis experiments? If we block early in the pathway versus late, when do we see this phenotype? And those are ongoing studies. I think the key thing that we need, and that this is what I alluded to earlier, is we're really viewing what's happening in these vesicles indirectly through the measurement of cytosolic heme. And I think the technology we've been trying to develop that'll be really enabling is actually to probe this in these acidic compartments and in the vacuole, and then revisit what's happening with heme in those compartments when we look at these mutants. So I think your point is an excellent one. I also think, as regarding your second point, I think absolutely heme has to find its way to enzymes that have to be heme loaded in the ER, in the secretory pathway. I think this is an important component of that. One thing that we haven't directly probed is what happens when these pathways are disrupted. What happens to the activity of cell surface heme proteins? We know that there are membrane-bound heme proteins that whose activities we can probe. We just haven't as yet. And the question is, are those hemolylated? And I think that's a really good point.
2 more questions.
Thank
Roy,
you.
and then Roland.
Hey, Amit, how's it going?
Hey.
So some years ago, Dennis's lab showed that isosome, at least particularly in yeast, would be important for heme transport. So how does that fit into your narrative?
Yeah, that's a good question. So we haven't seen any evidence for the isosome. I don't know how to say it. Isosome, icososome. I guess it's isosome. We haven't seen evidence for that from our screens. But I don't think we would have found evidence for our screens. I think the paper that I'm remembering, and you can correct me if I'm wrong, it was a screen that found a point mutant in that pathway that resulted in a phenotype that was observed. And here we're just looking at just deletion mutants. And as far as components of that pathway, those didn't come up in this screen. But I don't know that we've looked directly at it in a more rigorous and logical way.
Yeah, well, there was a gain of function mutation that they identified, but I'm just curious if that might be compensating in some of your mutants,
Yeah. We haven't directly looked at it, but the fact that it's a gain of function mutation would suggest why we wouldn't have seen it in our deletion screen.
And then Roland, Lil.
Yeah, Amit, simple question. Where would the heme in the endosomal compartment actually be? In the lipid membrane, as I would expect it, or would it be in the lumen as you drew it?
Yeah, that's a great question. The answer is I don't know. I think in principle, it could be either. We know that heme has the ability to partition into lipid membranes. I don't think I can rule out that possibility. I think that it's an open question, and I think it's one we have to figure out.
And
I think from a regulatory point of view, you might think that it should be in the lumen if there's active transport, between the lumen and the outside. Maybe there has to be an active transport mechanism. If it's in the membrane, just sort of hitching a ride. Well, I don't know. I guess I'm imposing my sort of human bias on this. I guess I wouldn't know what to think from a regulatory point of view.
Thanks.
Yeah.
One more question. Sorry, this longer break than I anticipated, but Dennis, go ahead and ask your question. Dennis Wingy.
Okay, so good to see you, Amit.
Hi, Dennis.
Is the mito-ER contact important to get heme in these secretory vesicles?
Yeah, that I'll address that point in just a couple of slides, Dennis. But I think the answer is yes. But genetically, it's been hard to figure out. But let me maybe if you give me a few slides, we can readdress that point.
Thank you.
Good. All right, so I'll keep going then. But these are all excellent questions. I'm really glad that I prepared a shorter talk than I would normally, because I think this is exactly what we have to do here, is have this discussion. So so real, the conclusion from the screen is, probably about 80% of the genes impact these, this Golgi vacuolar sorting. The other 20% are just sort of random metabolic enzymes. Okay, so things you would never have predicted to impact heme trafficking or metabolism just show up in our screen. I'll give you an example of one of those enzymes that we've already published on, but that might give you an idea as to these moonlighting roles for other metabolic enzymes in regulating heme availability. And so one of the things that we found very early on that was corroborated by Dennis Stewart's lab at Cleveland Clinic, but we also ended up working on this a little bit together also, was GAPDH as an enzyme that controls heme bioavailability. So yeast have three GAPDH isoforms. TDH3 is the most abundant isoform that's expressed under the growth conditions that we're employing in our screen. And when you delete it, you see a increase in heme bioavailability as registered by this decrease in heme sensor ratio. But what you also observe is that there's a decrease in the activation of a nuclear heme regulated transcription factor called HAP1. So this led us to think that GAPDH may be an important component of the cytosolic heme buffer, but it also plays additional roles in somehow regulating the transfer of heme to the nucleus, where it can activate HAP1 with heme. There are a lot of mechanistic questions there that we don't know the answers to. Working with Dennis Stewart, we were able to identify a potential heme binding site for GAPDH. And so it turns out that there's a conserved histidine at the protomer interface that can bind heme between GAPDH protomers. So GAPDH is normally a tetramer, and heme, we have evidence that heme binds to a key histidine residue between the different GAPDH protomers. And so this in it itself raises interesting questions. So GAPDH is typically a tetramer. Do you have to see GAPDH dissociation to lower oligomerization states to facilitate heme transfer? And what's the active heme transfer agent? Which protomers or oligomers may be relevant in the cytosol versus the nucleus in facilitating heme activation of HAP1. Dennis, in parallel work that we weren't involved in, has shown that GAPDH can also regulate heme transfer to nitric oxide synthases and other heme enzymes. So I think that there's some interesting roles for GAPDH here, independent of its role as in glycolysis and glucose oxidation. And we've identified a number of other enzymes that have the same sort of phenotype as GAPDH, that when you delete it, there's either an increase in heme or a decrease in heme. And there's basically many graduate theses worth of work to sort of figure out the roles of these other enzymes. So I'll leave that point there. We're also iterating these screens to look at other organelles. So Maddie Willoughby in the lab has been looking at factors that regulate nuclear heme availability in high throughput screens, as well as what regulates mitochondrial heme availability. And so these are ongoing screens that hopefully in the next year or so we can say more about as we complete the work. I'll sort of switch gears now to talk about heme trafficking dynamics. So far, I've just been telling you about what's happening with the steady state distribution of heme. I had a really talented graduate student who's at Utah in Adam Hughes's lab, Osiris Martinez Guzman, who developed this really interesting experimental paradigm where she designed an experiment to watch heme be trafficked to different organelles as it's being synthesized in living yeast cells. So the experiment is the following. So you can take our heme sensor, target it to different organelles. That's indicated by these light bulbs here, or these flashlights here. And And then you can inhibit heme synthesis using a heme synthetic inhibitor. And then you can remove that inhibitor and just watch the flow of heme to these different compartments as heme is being resynthesized. And when you do an experiment like this, you find that the hemylation of the heme sensor, the heme binding to the heme sensor, has a characteristic kinetic curves associated with the transfer of heme to the sensor as heme is being synthesized. So time zero is when we alleviate the block in heme synthesis. So there's a lag phase over the first 30 or 40 minutes. And once that lag phase is gone, there's a characteristic exponential phase where you see heme start to load the heme sensor in these different compartments, whether it's the cytosol, mitochondria, nucleus, and finally a saturation point where you've saturated the heme sensor with heme. Now, what's really interesting about this is, well, two things interesting about this is, one is that it appears that there are parallel routes of heme distribution. You know, I would have naively expected that heme populates the cytosol first, followed by, or the mitochondria first, followed by the cytosol and the nucleus. What this data is telling us that you have near equal rates of heme sensor population across compartments. Moreover, you have a slightly faster rate of heme binding to the nuclear heme sensor than even the cytosol or the mitochondria. And you can do this over many different cell types, over multiple experimental trials. And you see that on average, there's about a 25% increase in heme loading of the sensor, or the rate of heme loading of the sensor in the nucleus compared to other compartments. So this raised some questions as to how is heme getting the nucleus faster than other compartments. And this was alluded to by Dennis's question, but we suspected that maybe there are specific mechanisms for heme to access different compartments, including the nucleus. And one thing that we went after were organellar contact sites between the mitochondria, the ER, and the nucleus, and other contact sites. So I'll just briefly say that mitochondria are highly dynamic organelles, and they form contact sites with other subcellular compartments, including the ER. And so one of the things that we probed was, you know, if we disrupted ER contact sites and points at which we know ER constrict around the mitochondrial membrane, could we disrupt the flow of heme to the nucleus? So we tested known factors that impact mitochondrial ER contact sites, as well as factors that impact mitochondrial dynamics. And without extensively going through the data, I'll just say that it's a sort of a mixed result. So what we did find was that GTPases involved in mitochondrial fusion or fission are absolutely required for or regulate mitochondrial nuclear heme trafficking. So DNM1 is a GTPase involved in mitochondrial fission. So when you delete DNM1, there's a faster rate of heme trafficking to the nucleus, as indicated by this green trace. But when you delete MGM1, which is required for mitochondrial fusion, there's a suppressed rate of mitochondrial nuclear trafficking, as well as just generally lower nuclear heme levels in the mitochondria. But when you ablate both of them, as in this double mutant, you see that there is, wild type heme trafficking rates are restored. So, but when you delete so we know that factors like DNM1 and there's another GTPase that I'm not showing here, GEM1, that's involved in regulating mitochondrial ER contact sites, also impacts nuclear heme. But basically my point here is that these GTPases, whether it's MGM1 or DNM1 or GEM1, they impact mitochondrial nuclear heme trafficking. But when you perturb known components of mitochondrial ER contact sites, we don't see a phenotype. So you can ablate the MDMs, or let me go back to this slide. You can, I don't have a good slide here, but you can ablate known factors responsible for maintaining these mito-ER tethers, and you see that the rates of heme trafficking are not affected. So I guess the take home from this is the story is complicated, that in a way that I don't think just genetics is going to address. And so what we're trying to do now is target the sensors more locally to sites of to these contact sites, and try to get a more refined understanding of trafficking dynamics at these contact sites. As we were doing this work, more recently, people like Maya Schuldner and others have found that there could be mitochondrial nuclear contact sites. And so we're trying to test those mutants in those pathways as well to develop a fuller picture of this. So I think these interorganeller contact sites are going to be a key story here, but they're incredibly challenging to dissect genetically just because of pathway redundancy. We know that, for example, mitochondrial vacuolar contact sites are pretty dynamic and plastic. Mutations and other pathways can impact the prevalence of those contact sites. So I think that it's going to be a complicated question to tease, but I think at this point, the field has moved far enough ahead that we have the right sets of mutants to start doing these types of experiments in to better establish the role of these contact sites in regulating heme trafficking.
We have a question from the
Yeah,
group.
quick question. This is really, really fascinating. I'm sure I didn't get to read the paper you cited. I'm sure you did this, but have you done the control experiments where you've looked at the KD for heme binding of all of the different probes in a cell-free system? I'm just wondering that if the nuclear targeting sequence or whatever small modification you made to this protein that targets it to the nucleus subtly changed its KD for heme, and then you're getting this sort of change in baseline.
Yeah, it's a really good question. So I'll tell you that the tags don't make a difference in terms of the binding affinities. Well, I guess we never published that work, but it's or showed it in the paper. But that's it doesn't affect the affinities. But you're bringing up a really good broader point here, is that we're looking at really just the heme that's available to the sensor. So you can't necessarily infer that the rate of heme is being trafficked differently to these organelles. I guess more locally, you can just infer that the rate of heme binding to the sensor is different in these organelles. And then you can ask the question, well, what controls that rate? What's the rate limiting step of heme transfer to these different organelles? So each of these compartments are going to have their own heme buffering pools, components that can bind heme. And so I think one thing we'll discover if we do screens using this heme trafficking dynamics as a readout or steady state heme as a readout, is that we're going to come to appreciate that there's different components of this heme buffer in these different compartments. And ultimately, I think changes in that, or the composition of that, is what's governing these different rates of heme binding that we're observing. But it's a really good point. And I think that it's a really fascinating question that ultimately is going to get us to what the physiology behind these effects that we're seeing.
Thank you so much.
Okay, so I will move on to my last sort of vignette here, and I think I can do this in the last 5 minutes or so and still keep on time. So I've told you about approaches we've taken that look at steady state heme distribution, trafficking dynamics. The last point I'll leave you with is what are targets of heme signaling, and how are heme signals initiated and mobilized? And so a lot of this work is currently unpublished, but we hopefully are very close to publishing it. So I don't mind that it's on a recorded presentation, but we should talk more, I guess, if people have questions after I get through this last little vignette. One thing that we've noticed, since the beginning of setting up our lab and working on these projects, is that subcellular heme pools are highly dynamic. So we've found that a number of cellular perturbants, small molecules like nitric oxide, can dynamically mobilize heme within cells. So here's a very early experiment that we did that's since been corroborated in mammalian cells from Dennis Stewart's lab very recently. But basically, you can pulse cells with nitric oxide. You see these rapid mobilization of heme in the cytosol, the nucleus. indicating that this labile heme is a dynamic entity. We found that heavy metal stress and heat shock and other stressors can mobilize labile heme. You see these transient increases in chelatable heme, things like cell cycle progression, glycolytic metabolism, hydrogen sulfide. All of these are things that just mobilize labile heme. We see dynamic changes in sensor detected heme. So this naturally begs the question, what is binding to heme in response to these stimuli? What is releasing heme in response to the stimuli? Moreover, is there a physiological consequence to that? Is whatever is binding and releasing that heme actually have activity or functions regulated by this heme? And so these are the questions that I'll hit on just in the last couple of slides of my talk here. So in the last 2 years, or maybe longer actually, our lab has been working on trying to develop proteomics and chemoproteomics approaches towards identifying new heme trafficking and signaling factors, primarily with an eye towards what's binding and releasing heme in a dynamic fashion. And we've taken a multi-pronged strategy, but I'll just share with you one particular way we've gone about it in the interest of time. And all of this work is done in collaboration with the Torres lab in the School of Biology here at Georgia Tech, and the student pushing this is Hee-Jung Kim. So what we wanted to do is use mass spectrometry to identify proteins that dynamically bind and release heme, and I sort of call this a heme signaling network just to give this phenomenon a name that we can use to talk about it. So I refer to anything that binds and releases heme under a particular context, a heme signaling network. And we could debate with the name and the implications if we need, but that's just what I'm calling it. So the premise of this experiment is that in the cell, there's some distribution of hemoproteins or heme-regulated proteins between its holo and apo states. The idea is that a holo hemoprotein, one that has heme binding at whatever its heme binding site, a holo hemoprotein is going to interact less strongly with a heme agarose resin because its heme binding site is already populated. Whereas an apo hemoprotein will interact more strongly with a heme agarose resin because its heme binding site is less populated. What allows this experiment to work is the kinetics of heme exchange is relatively slow, such that you wouldn't see heme exchange between exogenous, you wouldn't see competition between heme agarose and endogenous heme at timescales in which we're doing this experiment. So that's a nuanced point that we can talk about later. But basically the premise of the experiment is what I'm describing here. And you can validate this using a number of model hemoproteins where you take an apo hemoprotein, you can see that you can pull it out using heme agarose, but if you load it with an equivalent of heme, you don't pull it out on heme agarose precisely because of the model that I'm showing here on the left of this slide. And so really the question is what are proteins, using this differential heme agarose affinity, what are proteins that bind and release heme in response to various stimuli, and can we identify it using mass spectrometry? And so the answer is yes, I think we can identify this using mass spectrometry. This is a snapshot of results that Hee-Jung Kim, along with David Hanna, discovered, where if you subject cells to heavy metal stress, this is a stressor that we know increases labile heme. We identify a group of proteins that, using this framework that I just described, can release heme in response to lead stress, and a group of proteins that acquire heme in response to lead stress. And these are the groups of proteins that we care most about as defining this stress responsive heme signaling network. And I'll just, in the last 2 minutes I have, I'll just give you a sense of what we're seeing here. We've found is that heme regulates, what the mass spectrometry experiments have told us is that heme regulates the ubiquitin proteasome pathway at a couple of different levels. We've found that one major target of heme signaling is the E1 ubiquitin ligase, UBA1. So the E1 ligase, what it does is it uses ATP to activate ubiquitin to then transfer it to downstream ubiquitin conjugating and conjugating enzymes and adapter proteins to ultimately ubiquitinate downstream targets. And what we found is that heme can regulate the activity of UBA1. So this is a ubiquitination assay where we're probing the ubiquitination of UBA1. So when UBA1 is activated, UBA1 will form a thioester linkage with ubiquitin, which we're reading out here in this experiment. So in the absence of heme, you can activate UBA1 with ubiquitin, and we see a band corresponding to ubiquitination of UBA1. When there's an equimolar presence of heme, you see complete inhibition of the ability of UBA1 to become activated with ubiquitin. So our results told us that not only does heavy metal stress increase heme loading of UBA1 during stress, but that has the consequence of inhibiting UBA1 activity. You can go through and do experiments to probe the specificity of heme regulation of UBA1. You can look at protoporphyrin 9 or zinc protoporphyrin 9, and those don't have the same effect as heme. With heme, you see an inhibition of UBA1 activation, whereas with protoporphyrin 9 and zinc protoporphyrin 9, you don't see that same inhibitory effect. You can just look at iron salts, ferrous and ferric iron salts, and see that it doesn't impact UBA1 regulation the same way that heme does. Lastly, you can also see that in vivo, heme regulates UBA1 activity. Here, this is an assay where we IP UBA1 and detect ubiquitination of UBA1 from the IP'd material. And you can see in heme depleted state, in heme depleted cells, and this is a concentration of succinyl acetone that inhibits heme synthesis to a level where we completely deplete labile heme, but only modestly deplete total heme levels. So this is a modest dose of SA. And we see that there's an increase in UBA1 activation in cells as read out by this assay here. So basically, I won't, so let me just skip ahead to the punchline here. So basically, what we see is that stress, in the combination of the proteomics and some of the biochemical data I've showed you, is in response to stress, heme binds to and inhibits UBA1 activity, and this has the consequence of preventing ubiquitination of proteins in response to stress. And the physiological benefit of this, we think, and this is a work in progress, is that we know that ubiquitin-independent pathways are important in the stress response. And this is because misfolded proteins, damaged proteins, oxidatively damaged proteins are degraded by the 20S proteasome in the absence of ubiquitin. And so the underlying context of this mechanism is that maybe in response to stress, there's heme binds to UBA1 to inhibit UBA1 activity to promote this ubiquitin-independent pathway of proteasomal degradation. We think this could be a benefit for stressed cells. And this is not to say that all UBA1 activity is depleted. You still have some active UBA1, but so we think that heme could be the stress signal that helps cells shift the regulation of ubiquitin-dependent versus independent pathways for protein degradation. I'm out of time here, so I'm just going to stop here, and I'm happy to take any questions regarding any aspect of my talk. And I'll just thank the people that did the work. Much of the work that I'm talking about here was spearheaded by Osiris in the middle, David, Hugh Jung, and work that I couldn't tell you about. Well, I'll stop there. Contributions from lots of lab members, and of course, funding from the NIH and NSF. So thank you all.
Great, thank you. That was a great talk. Really, actually, very thought-provoking as a person who's a cell biologist who thinks about membrane trafficking often. I want to invite, I think Dennis had one question, and I'll invite him to ask that question first.
Amit, did you, in your mutant screen, find any flipases that could mediate translocation through the IMS?
Oh yeah, it's a good question. I don't think so. I think the answer is no, but now that you say that, I'm gonna go look back at the data and just look for that. But that's a really good question and point.
Paul, go ahead.
Hey, Amit, I know we talked about this before, but I guess what's your current thinking of how you interpret the sort of effective concentrations you get from these sensors in different compartments? I'm curious if you've gone to, higher resolution approaches. I don't know how much. I'm curious how much heterogeneity you see in sort of ratiometric loading of your sensors distributed throughout, say, the cytoplasm or distinct areas of the mitochondrion or other compartments.
Yeah, it's a really good question. It is highly heterogeneous. So let me unpack that a little bit. So we haven't done super high resolution imaging as yet on this. But even with when you look across whether it's mammalian cells or yeast cells, across a population of cells, you definitely don't see homogeneous, homogeneous response from the sensor. Some cells appear to have more localized heme, closer to the membrane and periphery. Some don't. We're trying to better understand the basis for that. Even within a population of cells, cells are very different. Each cell will have its own unique distribution. I don't know what the underlying basis for that is. Part of this, I suspect, is because of cell cycle or metabolic cycling. We haven't really carefully controlled in a lot of these experiments cell cycle effects, and we already know that that cell cycle progression will alter labile heme pools. So I think that if we revisit some of those experiments and synchronize cells, regardless of what cell type we're looking at, we might begin to tease apart the question that you're asking.
Thanks.
Any other questions?
Yeah, I have another one. So is it known whether ferrochelatase shows any submitochondrial enrichment in kind of focal areas?
Yeah. So as far as I know about ferrochelatase, I think it's primarily membrane bound to the inner membrane. As far as anything further than that, I don't know. I know that there have been reports of ferrochelatase being found in I guess I don't know what you'd call them, like microvesicular compartments associated with like with microsomes. I don't know how physiological that is. So I guess I don't really know more about ferrochelatase localization beyond just it's present in the inner membrane. I don't know about the any heterogeneity in its distribution across the inner membrane and anything like that.
Thanks.
Other questions? You're welcome to unmute yourself and ask. I don't see anything in the chat currently, so we could be on time.
Nice.
Which is nice, but also it's very nice to see everybody and to talk. So that seems like a great opportunity for us in this weird time. But I want to thank you so much for your presentation, and I look forward to hearing more about these heme sensors. I was thinking on some of your assays where you're pulling on GAPDH or anything like that. There's a core here at the University of Utah that runs that does a protein metabolite interaction core, and I wondered if you had enough purified proteins, if you could see heme or heme breakdown products or heme precursors regulating the some of those proteins, even like GAPDH in its activities. That was just one thought I had.
Yeah, that's a really good point, Diane. So we, Hyojung Kim in the lab, developed a very sensitive assay to detect heme association with proteins. I didn't get a chance to show you that data here, but basically it's an IP of a protein, but then you're using a very well-known porphyrin fluorescence assay where you just boil the protein in acid and detect porphyrin fluorescence. And this gives us very sensitive detection limits where in standard IP sort of material, you can look for heme association. And this is a method I think we'll publish on soon, but relies on well published methodology already. All we're doing is applying it to IP material. But the question then is which of these proteins I've talked about can you find actually interact with heme in vivo using this IP assay? And you can extend it to some of the things that you were talking about. What other metabolites interact with these proteins, heme catabolic products, and so on.
Right. It's really fun to think about. Who knew?
Yeah.
Really, I mean, a simple vision for myself, but it's becoming, as expected, much more complicated, but cool, really cool.
Amit, I have one final question. If you were to take one of your sensors and tether it to the outer mitochondrial membrane and now do that kinetic experiment, would that light up early, or is it? Yeah.
Yeah, Dennis, that's a great question. That's exactly what I want to do. I want to get very focused resolution on this. Put these sensors on the mitochondrial inner membrane, outer membrane, ER mito contact sites, ER mito nuclear contact sites. And I think that's going to be very telling of the mechanisms of heme trafficking. I suspect there's going to be kinetic barriers for free exchange between these micro foci, and so I think that having that resolution and then screening for determinants of what regulates availability and rates at these specific locations will be telling of what is handing off heme from ferrochelatase to each of these, I guess, I don't know what you call them, micro compartments or micro locations. The other added point I can make here is that, Amy Medlock and Harry Daly have done some really nice work that showed that this heme biosynthetic enzymes exist in this multi-protein complex. And so heme has to be transferred within this complex to wherever it needs to go. And I think if we iterated the screen for the mitochondria as well as these sublocations, we can figure out what else could be interacting with this super complex to sort of gate the movement of heme. And I think those are the next year or two's exciting experiments on the horizon. It's a really good point.
Yeah, great job.
Great. Okay, we'll stop there. I want to thank everybody for their attendance and remind you that we have another seminar next month, which will be Susan Clunan. Hope you'll join us again. And Amit, thank you so much for your talk and for teaching us. I noticed on your CV that you have all these teaching awards, and I'm thinking, well, I just got taught very well again. So thanks so much.
I appreciate that. This was a lot of fun. Thank you so much for hosting this and allowing us to share our latest. I appreciate that.
Yeah.
Good seeing everybody.
All right, everybody take care.
Bye, everyone.
Yeah, bye.
Thanks for dropping in.